the data we presented is a very large phase 1 trial with a monoclonal antibody that blocks a compound, let's say an antigen that is called PDL1. PDL1 is, if you want, a diplomatic immunity that is used by the cancer cells to avoid their destruction by infiltrating activated T cells. If you want, when you have a cancer cell, the cancer cells, when they die, they release antigens that in theory will induce an immune response that will activate T cells that should be able to destroy the remaining cancer cells. But in reality, these cancer cells avoid the destruction by upregulating and expressing either themselves or their microenvironment PDL1 that will prevent the activity of the immune infiltrating cell. So it works like a diplomatic immunity. You're a bad guy, the police comes in, wants to take you, but you have a diplomatic passport and says, go back. So an anti-PDL1 is a compound that will take away the diplomatic immunity and allow the cops, the immune cells, to do their job and take the bad guys away. By blocking PDL1, you will block two interactions that are important. The PDL1-PD1 interaction and the PDL1-B7-1 interaction that will allow not only T-cell priming, but also the restoration of a strong anti-immune response. So your own lymphocytes will be doing the job of fighting the cancer cells. Well, in reality, this was a study, very large phase 1 studies, as many of the ongoing phase 1 studies today that have hundreds of patients. So we have left the era where phase 1 was about 30 patients and only toxicity. We are in an era where phase 1s are hundreds of patients, also for activity. In that study, we deliver the MPDL3280A, well, that's a long name, let's call it MPDL for monoclonal PDL, IV as a monotherapy every 3 weeks, up to 1 year of therapy. And this was given to many different tumor types: breast, colorectal, gastric, melanoma, renal cell carcinoma, non-small cell lung cancer, lymphomas. And in every single one of these tumor types, objective responses were seen. The data I presented was specifically focused on the 85 non-small cell lung cancers that were enrolled in that trial. In that population, which was heavily pretreated because over 55% of those metastatic non-small cell lung cancers got at least 3 lines of previous therapy. Well, in that population, the global objective response rate was 23%. That's very significant when you are on 4th line and beyond of therapy. Phase 1, as I told you, is not only about toxicity. That is a 20th century definition. In the 21st century, phase 1 is both about defining toxicity and acquiring data regarding activity. So you really need to change your paradigm or your software, okay? But indeed, toxicity was carefully and thoroughly evaluated in this trial, and the profile of toxicity of MPDL was extremely favorable, mainly grade 1 and 2 adverse events with no need for an intervention. No pneumonitis, this is important because PD-1 antibodies have been related to pneumonitis. We saw none. 1 single immune-related severe adverse events with diabetes. So a good safety profile. We know that lung tumors are among those solid tumors with the highest mutational load in the tumor cells. And we also know that smokers among lung cancers have more mutations than never smokers. So we hypothesize that maybe the mutational load will be related to the immunogenicity of the tumor and therefore the activity of the compound. And indeed, we had 80% of current or former smokers and only 20% of never smokers. But the response rate in the current former smokers was higher, 26% as compared to 10% in the never smokers. And this is the first time on the last 10 years that a molecular targeted agent brings more hope for smokers than from never smokers. I'm not saying you should keep smoking. the best you can do is to stop smoking. But if you are lung cancer that has arisen from smoking, usually most of the innovation, the tyrosine kinase inhibitors for EGFR mutation, ALK, and others, were only for the never smokers, where most of the oncogenic mutations happen. Here we have great news. We have the smoking people who have the possibility to benefit from a new compound with mild toxicity and clear activity. Well, as I said, I have been a strong member of the immunoskeptical clinicians. Those who have seen over the last 15 years huge investments in immunology, at least in the lung cancer setting, with zero results. But this is clearly a game changer. There is no waiving the data here. Really, whether it is PD-1 data already presented with compounds from BMS or this PD-L1 antibody from Genentech, and we look forward today's presentation of the PD-L1 from MedImmune and the PD-1 from Merck that will be presented in World Lung Cancer, these compounds have as monotherapies. So it's a single IV shot of an hour, correct? In outpatient setting, every 3 weeks, can change your life. A quarter of the patients will have a response. And on top of that, in our trial, we demonstrated that the level of expression of the target, PD-L1, in the tumor immune infiltrating cells was predicted for higher efficacy. The higher the level of PD-L1, the higher the response rate. So this is really exciting times. When you have something that is so powerful, so well tolerated, you want to partner it with other important cornerstones of anti-cancer therapy. Obviously, combining it with other targeted agents makes a lot of sense. I personally believe that combination with oncogene kinase inhibitors like EGFR tyrosine kinase inhibitors or ALK inhibitors is very important. On the other hand, obviously, chemotherapy remains the cornerstone of therapy in systemic stage 4 non-small cell lung cancer. And there are combinations that are being tested. But you need to bear in mind the fact that when you combine an immune checkpoint modulator like a PD-L1 or PD-1 antibody with chemotherapy, one of the problems is that chemotherapy needs steroids. Otherwise, you vomit too much and you have too much side effects. Well, steroids are an antidote to building an anti-immune response. But that's going to be the challenge of the future. Well, I think the good news is that in non-small cell lung cancer, in smokers and notably in squamous, we have a very promising compound. These compounds are not commercially available yet. So clinicians who have patients in good performance status 0, 1, metastatic who fail standard of care should be referring their patients to the cancer centers or university hospitals that are currently testing these compounds through specific clinical trials. Well, I cannot comment since I do not develop PD-L1 for Genentech. I'm an investigator from an academic center, but I am sure that beyond non-small cell lung cancer, as you know, there is already a huge development in the melanoma world, where not only PD-1 and PD-L1, but also their combination have already been tested. In renal cell carcinoma, there are huge investments. Question mark whether the pharma is going to go full speed beyond these 3 tumor types, melanoma, renal, and lung, to other tumor types where some responses have been seen. Immunology really works in lung cancer. This is new. This is changing the paradigm. We used to consider that these were, very expensive initiatives with zero results. Now we know that these initiatives can lead to 25% objective response rate as a monotherapy. That's high. So immunology has entered the thoracic world. Immunology is working. Immunology brings promise.